Okay, thank you so much. So thanks everyone for joining us, and it is earlier than usual, so I appreciate everyone making the change in their schedule. I know many of you look forward to connecting this way socially when we're sort of physically disconnected from each other. Hopefully everyone is staying safe and doing well. So it brings me great pleasure today to introduce to you Roger Barker, Professor Roger Barker, who was suggested to us by Dr. Simon Lewis, who you may remember from the gait and freezing talk about a month or two ago, which has been a very popular episode. You should definitely watch it since he's highlighted he's very funny, and he roasted one of our other colleagues, Bas Blom. So it's really fun to connect these dots internationally and see where everybody's trained. But Simon told us about his training and where he really learned about Parkinson's disease and has gone on to really inspire people in Australia. And that was from Professor Barker. So I think they did some research together. Simon ended up seeing a number of patients with you, or some of your patients, I guess, there. Maybe you can tell us a little bit about your collaboration. So Professor Barker is a professor of clinical neuroscience and an honorary consultant in neurology at the University of Cambridge. He's trained basically at, you name it, at the most prestigious institutions in the UK. He spent some time at Oxford and is now at Cambridge, and is doing some really amazing work in a number of research areas. He's really focused on Parkinson's, and he also does some inspirational work in Huntington's disease as well. A lot of patients have had a lot of questions about such cell-based therapy, and I really wanted to get the world expert on here to kind of dispel some of the myths, to help us understand why it's important to seek out these therapies only through certain channels. And so Professor Barker will enlighten us with some slides first. Before we go there, I wanted to learn a little bit about how you got into this field in the first place, maybe how you got into medicine, why you chose medicine, why neurology, and then teach us a little bit about why you specifically have chosen this area of Parkinson's research and beyond.
Thank you very much, and it was very kind of Simon to recommend me. Yes, Simon was my very first clinical fellow. There were three of them, Simon, Tim and Tom. So Simon was very easy to remember because Tim and Tom I used to get confused. So, yeah, I got into medicine a bit by chance, to be honest. When I was at school, everyone I seemed to know wanted to do medicine. So I thought, why not do that? I've always very much enjoyed trying to help people. I like the challenge of trying to solve problems. And so that's the reason I got into medicine. And then when I was at Oxford in my second year, there was a chap, the late Tom Powell, who was one of the great neuroanatomists in the late 20th century, who made neuro seem so easy that I thought, this is what I need to do. And also the challenges for therapy, I thought, were way behind anything else. So, in heart, quite interesting, but at the end of the day, if it all fails, stick a new one in. Whereas with the brain, I thought there were many challenges. So I got very interested in neuroscience and neurology. I then completed my training, did neurology, hated it. Thought, why am I interested? I did it at Queen Square, one of the big places in London, and we spent ages deciding whether someone's got PD, PSP, MSA, DLB, all these letters, and I thought, what does it really matter? Anyway, 6 months later, I thought it was actually very interesting to know whether people had PD, MSA, PSP, DLB, and that set me on my course in research, and I started my research in I've researched all my life, but really my PhD in 91 in Cambridge on trying to repair the brain with cells. And that's been a large area of interest since then.
Wow. So really 3 decades almost of doing this research.
Long time. I'm very old.
Well, I will share with you one story that Simon told me before we move on. And I think this is something that we actually share. So I asked Simon if there was a funny story about you. And he said that you don't like to travel, apparently. You spend as little time on the road, which is the opposite to some of us. But when you do travel, you take everything in a backpack, which I actually am known to do as well. And apparently you were gifted something, maybe you could tell us about this, in one of the Asian countries that you then had to wait for on the baggage claim area. Do you remember this story?
I don't remember that. I remember going to carrying everything in a small knapsack, and we went to give some talks. A group of us went to Japan, and they were very honoured for us to go there, and so gave us all little samurai swords as a present to thank us for going there, which of course then created a major problem because little samurai swords on hand luggage these days don't take you very far through security. Fortunately, we managed to find somebody else who had a massive suitcase that they were bringing back, so we shoved them all in that.
Oh, okay. He made it sound like you were waiting for the samurai sword in the baggage claim by itself.
Oh, no,
But
yeah,
that is very cute. I am definitely travel lightly as well, and am missing traveling. So that's very interesting. So maybe then, Professor Barker, you can start with your slides and maybe teach us a little bit about, sort of what you have sort of gained knowledge around with these cell-based therapies, and then we can open it up to some questions. And I have some questions as well, since it's such a hot topic.
Yeah, sure. So I'm just going to share my screen.
And in the meantime, somebody's written, "You look young, like a fellow." Something they said that you look too young to be having been in medicine that long and researching.
Yeah, well, so. I think it's very kind of them, but they may need to go to their opticians and check their eyes. So yeah, thanks very much. So what I thought I would do in this little talk as an introduction is to sort of say what are we trying to achieve with cell-based therapies, and then a little bit about how we started, which is actually with Simon, as it happens, about trying to define subtypes of disease in which to match the therapies. And then I was briefly just going to walk through what people have shown with cell-based therapies, both historically and where we are now, and also some of the problems, and then end up with what can we look forward to in the future. So the real sort of slightly sci-fi type approach. So I think the most important thing, so I'll do this as a series of questions. So the first question I think one needs to ask is, what are we trying to repair in Parkinson's? What is this all about? Because one of the key things when people talk about cells is they sort of talk about cures and getting rid of Parkinson's disease. And really, they're not cures. They're designed to treat a cardinal feature of the condition. So what we're really trying to do is to repair one of the core elements, namely the loss of these dopamine pathways from the top of the brainstem in the nigra up into the striatum, these nigrostriatal pathways, which we know are critical to Parkinson's disease, because if you give people dopamine drugs, they do very well. But obviously, if we're going to try and repair that particular pathway, there are essentially only two ways to do it. But importantly, and this is what I want to get across, is we're not going to cure it, because the disease process, whatever that is, with alpha-synuclein and such like, will continue. What we're trying to do is, in effect, a repair of a specific network, with the hope that with that, we can actually make people much better. So how could you basically repair that? Well, you don't have to be Einstein to work this out. There's two ways you can do it. It's essentially the same as if you have a flower bed with not very much in it. So if you want to grow roses and you've got rather weedy roses there, which would be, if you like, the loss of the dopamine cells in Parkinson's, you can either add fertilizer and grow the roses that you already have there. And that's growth factors, which I'm not going to be talking about today, but I'm happy to take some questions on it. So there are various sets of letters of growth factors, particularly GTNF is the one most people would have heard of. So that's putting fertilizer onto a failing dopamine system to make it grow. The second is to say, I'm not really going to grow the roses I've got there. They're all a waste of time. I'm just going to replace them. And that is replacing the specific dopamine cells which are lost in Parkinson's disease. And that's what I'm going to be talking about today. So the idea here is in Parkinson's, you lose half of your dopamine cells, which is only 250,000 dopamine cells on either side of your brain. And when you lose those, you start to develop the motor features of Parkinson's. And we want to put those back. So that's the question we're going to ask. That's the challenge we faced. And that was the problem that arose when we first started thinking about this 30 odd years ago. So the first question takes us back actually to a sort of more fundamental question is, well, who are we going to treat with this? So I think the idea that Parkinson's is one disease entity, everyone has the same disorder, everyone looks exactly the same. We all know that's not true. So the very first piece of work we did to try and use or try and think how we could take cell therapies back to the clinic or into the clinic for people with Parkinson's was to ask a question about actually how many different types of Parkinson's are there, and is there a particular group of patients that would do very well with this therapy? And so this was a study we set up at the beginning of the century where we collected everybody within the county of Cambridgeshire in the east of England, and we followed these people to the present day. So we've now been following these people for 20 years. And so this has enabled us to come up with a whole series of bits of information about natural history of Parkinson's. So, for example, average age to get Parkinson's is 70. The death rate with people with Parkinson's is no different to the death rate in the population. So it does not shorten people's lives. If you look at death certificates, most people die with Parkinson's disease, not of Parkinson's. So there's lots of information we've learned about natural history, But what we were particularly interested in is if you follow people over time, do they all follow the same course? So 20 years of pretty much everyone in the same place, or have people ended up in different areas and different types of disease? And this led us to sort of come up with this idea that there are two main types of Parkinson's disease, or they're really diseases that sit at the end of a spectrum. The first are people who are younger patients, may have some subtle thinking problems, but actually have a disease which is very much concentrated around the dopaminergic loss. That's what dominates their disease, very responsive to medication. In contrast, there's a group of older patients, typically presenting over the age of 72, who have problems with certain thinking tasks, who unfortunately early on develop major problems and go on to develop an early dementia. Now, why is this important for our types of therapy? And what determines where people go? Well, obviously, age determines that. There are some genetic variations. And interestingly, levels of inflammation that people mount in response to the protein may be very important in this, which opens up potentials around anti-inflammatories, which, again, I'm not talking about. The key thing here is if we don't take this into account and we just apply cell-based therapies around dopamine, as I've said, to this group of patients as a whole, you can see what can go wrong. So I take 10 patients here, 5 with the young onset, all around dopamine, 5 with older onset, problems all over the brain, develop an early dementia, don't have a disease which is only around the dopamine system. I ask at 2 or 3 years, which is the end of my study, are you better? The younger group say, terrific, fantastic, those dopamine cell therapies have made me normal. The other 5 patients have developed a dementia. The cell therapies haven't helped. In fact, I'm worse for the cell therapists. Therefore, we've had a negative impact. Put the two together, makes no difference. Bin the treatment. Let's move on to the next one. And I'll come back to that because that has been a problem in this field, that we haven't selected people carefully enough to decide how to treat them. So this group on the right, whilst you might think the group with the older group may be the worst group for our cell therapies, they are in fact the absolute best group for any disease modifying therapies around drugs and such like to slow down the disease process. But I'm going to be concentrating about this other group who we think actually are the better group. So we've decided what we're going to do. We're going to repair the dopamine system in the Parkinson's brain. We're going to do that by putting back dopamine cells, and we're going to do it in this particular group of patients. So how close have we come to doing that? How successful have we been with this approach? So there are lots of different dopamine cells you could consider to be used. So the first place people started in the 1980s was to reach around the body, try and find some dopamine cells in somebody's body, yank it out and shove it in the brain. And so there were various places where people found them. They did exactly that, and it didn't work. Basically because they don't really produce dopamine and they don't make dopamine nerve cells. So then the next question is, well, if we can't use dopamine cells from within our own body, where else can we go and get them from? Well, one is you can collect them from aborted fetal tissue. So women who decide to terminate the pregnancy, can we collect the fetal tissue, take out the developing dopamine cells and use that? Could we make dopamine cells from embryonic stem cells? These are these embryos from the IVF programs, which aren't used for reimplantation. Could we make dopamine cells from those? And then the third area is, can we actually take adult cells, so adult skin cells, adult blood cells, turn them into stem cells and then turn them into dopamine cells? These are the so-called induced pluripotent stem cells. And I'm going to go through each of these in turn. So where have we got to with this? Well, actually, if you look at fetal dopamine cells, so this is work which began in the 1980s, primarily in Lund in Sweden, and was continued during the 1990s. Then what was discovered, if you take out these developing dopamine cells from aborted fetuses, so the idea here is pregnancy is terminated, you dissect out the bit of brain where the dopamine cells are developing, which will give rise to the normal dopamine system, nigrostriatal dopamine system. You take them out, you turn them into a soup, you inject them into the brain. What was found? Well, actually, there were major long-term clinical benefits. Now, you can't really see this graph, and it's not that important. And if I can get this video to work, which I don't know whether it will on this, but we'll try. Here we go. So this is a video. Well, it was a video. It's just stopped now. But here we go. So this is a chap that was treated in Lund in the late 1980s. He's been followed up by my colleague Tom Folseny in London. This is a gentleman who's had Parkinson's disease at this point for 35 years. He's had a transplant for 20 years. And he's currently, or 25 years, and he's currently on no medication for his Parkinson's disease. So I think, whilst there will be skeptics around this whole approach, it's pretty hard to get away from the fact that when it works extremely well. So that is the clinical benefit you can see. Now, it's also been shown in other patients from that series. So here is a patient who was grafted in 1989. He died in his late 80s in 2013. And at post-mortem, what you see in that brown smudge are all the transplanted cells which have survived 25 years, more or less, after they're put in. So patients do well long-term with these fetal dopamine cells. They have surviving cells. They improve not just in terms of their motor features, but quality of life and non-motor aspects of the disease, which are very predominant now and well recognized in Parkinson's disease. And these cells release dopamine in a way that you'd expect. So when you stimulate the cells, they actually release dopamine, just like the normal dopamine cells in your brain. So all of this is very exciting because it seemed to be working very well in the 80s and 90s. Then there were two big trials done in America, funded by the National Institute of Health, both of which failed to show that they worked. So people in 2003 thought that this field was dead. Didn't work, deep brain stimulation works, why bother? So we went back and tried to look at the data and say, well, actually, is that right? Is the field not really worth pursuing because it's not any value? What can we learn from the trials that have been taking place today? And what you discover is actually it becomes rather difficult to make any firm conclusions. So if you look at the two studies from America, which are these two columns on the left, and the studies done in Lund, then these trials were absolutely identical in everything they did, apart from the type of patient they chose, the number of cells they put in, the way in which they were delivered, the extent to which the rejection process was switched off in the host, the end point for the trial, and the length of time they followed up. Apart from that, they were identical. So you can see that it's almost impossible to make any sense of this. We analysed it as best we could, and we decided that actually those people who would do best with this therapy, based on the limited information we had, were younger patients with a standardised way to put the cells in, with long duration of immunosuppression, stop the cells being rejected, and long-term follow-up. And that led us to do this trial, which we finished a couple of years ago, where we took patients with younger onset, earlier disease, and transplanted them with fetal dopamine cells, both in Cambridge and in Lund. We started in 2015 and we finished in 2018 with that study, and the end point will be next year. So that will be interesting to know what we've shown with fetal dopamine cells. Now, this was always seen, this trial, as a stepping stone stem cell transplant, and to highlight this point, in the 21 patients we grafted, 10 had 2 transplants, 1 had 1 transplant. For the 21 patients we transplanted, 87 other surgical slots were cancelled because we didn't have enough tissue to transplant. So logistically, we just can't take this forward. So we always knew that, but this was to try and prove that we could do better if we were more selective about how we took on this trial. And this led us on to trying to develop stem cell therapies. Now, there are many different types of stem cells that are out there. There are those which, as I say, come from the developing embryonic stem cells, and there are those which are the adult ones, which are made from adult cells, so-called induced pluripotent stem cells. So these are the 2 cells which we think are the ones which are likely to go to clinic, and in fact are already in clinic in some places. And you may already have heard about a recent paper that came out last month on this that I'll briefly touch upon. Now, in order to try and avoid some of the problems that we had with the fetal tissue and the way the field's gone, we decided that we would actually try and bring us all together into a more coordinated group. So this is the wonderfully entitled G-Force PD. This is us on a lawn in Cambridge with the River Cam behind us. We meet every year and have done for 7 years, except this year when we were supposed to meet in New York in April, and our friend COVID-19 got in the way of that. But the idea here is we have groups from Europe, groups from America, and groups from Japan and China that come together every year to exchange information on how we are progressing with taking stem cells into the clinic. And this group has actually also spurred on huge investment from companies in this field. So when we started the Eurotransplant program in 2015, world investment in stem cells for Parkinson's was approximately $0. By the time we transplanted our last patient in March of 2018, world investment in stem cells for Parkinson's was in excess of $1 billion. So this has become big business. So the groups that are working on this is ourselves in Europe, myself and Marlin Palmer in Sweden, and Novo Nordisk, the fourth biggest pharma company in the world, are now developing a cell-based therapy for Parkinson's disease. Lorenz Studer in New York set up a company called BlueRock, which has now been bought by Bayer, who are trying to develop a stem cell dopamine therapy for Parkinson's disease. Fujifilm funds Cellular Dynamics around Chicago to do the same. Aspen Neuroscience are a company which is recently come into existence, which are looking to take iPS cells into patients. In Japan, they've already started with a big funded program to do this in patients. China has several trials that are ongoing, and there's a trial knocking around in Melbourne, which is interesting, which I'll come to at the end. So this has become a big area. So where are we? Well, in terms of iPS, so this is where you take adult cells, turn them into dopamine cells and transplant them in. This group in Japan, led by Jun Takahashi, had this very important paper showing it worked very well in monkeys when you took human Parkinson's skin cells, turned them into dopamine cells, transplanted, they worked very well. And they started a trial in 2018 where they had a patient where they grafted one side of the brain, and then in June of last year, just before the World Parkinson's Congress in Kyoto, they transplanted the other side of the brain. So a patient in Japan has had an iPS-derived dopamine cell transplant for their Parkinson's disease. We don't know how well they've done, but we know that there's been no complications from that. More recently, as I said, there was this rather curious paper that appeared in the top medical journal, the New England Journal of Medicine, where a patient had their skin cells taken, they were converted into iPS cells, they were converted into dopamine cells, and they were transplanted into animals to show that they survived, which was of no interest to the patient whatsoever because they wanted them put into their brain, and they were grafted into that patient's brain. So this is the patient's skin cells turned into iPS cells, turned into dopamine cells, put into their brain. And what they found was that the patient tolerated them very well. There wasn't much change on the scan. There wasn't much change in their clinical score, but they certainly felt a lot better based on the PDQ-39. There are lots of issues around this, which I'm happy to discuss. It proved it was safe. I think there are very odd aspects to this particular patient's trial. But nevertheless, it shows that these cells survive and work. Embryonic stem cells, so these are the ones made from spare embryos. There are many companies investing in this. We now have good protocols where we can make lots of these cells. They work extremely well, and we are pushing towards a clinical trial, which we hope will start in the summer of next year. We're still going through all the regulatory process, but we have the cells in the freezer. We have patients from TransEuro who were not grafted lined up for the transplant. So we are moving towards that, and various other groups are ready to go, including the group in New York, as well as groups in China. The final thing I want to say just before I conclude is a word of caution. So there is a stem cell company called International Stem Cell Corporation who have made their own who've made their own stem cell line, which are from non-fertilized eggs, which are ethically more neutral because you destroy no embryo in order to make it. That's their main claim. They have now proceeded to transplant many patients in Melbourne in Australia with this. I think it's 12, 16. Their preclinical data shows that the cells really don't survive very well. So this is just a picture showing the one dopamine cell in the several million they grafted. When they treated their monkeys with this, so basically on this graph, they took monkeys, they made the monkeys Parkinsonian, and they grafted them. So if you look at the lines, over time, the group that received the low dose of their cells got better over time, which is very encouraging. Bit peculiar because the high dose group didn't do quite so well, but actually if you did absolutely nothing whatsoever to these monkeys, they actually got better on their own. So this is a bit of a worry because there's no evidence that these cells actually are really surviving, and that the benefits you see probably are not being mediated by the cells. The animals are just getting better on their own. But nevertheless, they're pushing on with this. And then there are groups in China which have done this with embryonic stem cells as well as these parthenogenetic cells. And these are the type of transplants they're seeing preclinically, which probably is not what you'd want in your brain if you had such a transplant. So just to conclude on this brief introduction, I think we can say that cell therapies for disease have a rational basis around dopamine cell replacement. They're not a cure. They're only trying to replace one part of it, but it's a key part. We have to be careful about who we select in the first instance, but we think we've identified the relevant groups. Fetal dopamine cells have produced mixed results, but when they work well, they work extremely well, by which I mean long-term clinical benefit over decades with evidence of surviving cells and improvement across many different paradigms. Stem cell therapies are now in the clinic. So there are at least two well, the iPS cells are now in both Japan and in the United States, but it's easy for all this to go pear-shaped if people don't do things following proper scientific rigor, and that's what G-Force PD is there to try and foster. And in the future, I guess, you sort of say, well, where will this take us? So what does this actually mean if this was to work? So if this worked, so if we thought these dopamine cell-based therapies could work, I say they're not a cure, but what would it mean for you now being treated with your Parkinson's disease? Well, this is a sort of fairly typical regime of what we use in the UK. So these are all the different types of drugs: selegiline and rasagiline, dopamine agonists like pramipexole, mirapexim, ropinirole. Then you get complication, a bit of amantadine, apomorphine, deep brain stimulation, duodopa. So what could a stem cell dopamine therapy do if it really worked? Well, it would look like that. So basically, you would have a stem cell therapy at the point of diagnosis, and you wouldn't need anything else. So everything we do for Parkinson's nowadays is related to the dopamine system or the complications of treating it. So everything we do in Parkinson's would be made redundant if these therapies worked. And beyond that, if all of this works, then why bother making a cell and putting it in your brain? Why not just reprogram your brain to make the dopamine cells you're missing? So the idea here would be to inject in viruses with the factors that we use in the lab to make dopamine cells to convert a population of cells you don't really need in your brain into dopamine cells and repair yourself. So it has huge potential, this field. It's still very much in its infancy. There are problems as we go forward where things could be derailed for various reasons. There are huge social aspects and ethical implications from much of this work, which people may want to touch upon. And finally, I just want to thank all the people who've helped me with the work over the years. This is my group and Caroline Williams-Gray group in a rather cold March. All the people who support us and our wonderful collaborators, particularly G-Force PD and the people in London. So I'm going to stop there. I'm going to stop share, and then I'm happy to take any, well, not any question, but most questions.
That was so amazing. I've never seen a talk so clear, really talking about the flowers and the weeds and all that kind of thing. And, you know, you've sort of meandered through the history of this very beautifully. I had watched a number of your other videos that are out, and I encourage people to watch these. There was a recent one with Patrick Grundon on inflammation that was very interesting, and there's just so much, you know, out there in the field of Parkinson's that's exciting and show therapeutic benefit possibilities. But also, I think the beauty of your talk is that it also comes with, you know, a grain of salt. And so, you know, just making sure that we're doing things safely and learning about the process in a collaborative way. So I really commend you for putting together G-Force PD and you know, working so systematically, because I think it is an area where there's a lot of hype and a lot of, you know, patients that are very excited to sign up for these trials. You know, it becomes sort of one of these things where as clinicians, we're trying to, you know, sort of give hope, but also be protecting our patients as well. So I think it's just important. A couple of questions. So it sounds like, just to break it down a little bit, and you can tell me a little bit about if I'm confused. So it sounds like the fetal cells are a non-go because it takes so much fetal cell volume to be able to transplant, and it becomes hard to even have enough, even though there's a few patients that have done well. Moving forward, it just is too difficult to just get the number of fetuses together to get, so that's the probably a kind of dead in the water. These other two approaches with taking embryonic cells seems like the thing that you're the most excited about, because it sounds like, number one, when we use the patient's own cells to try to, you know, make cells from skin, for example, and we have to kind of go back to, you know, making these cells less mature and make them pluripotent. So we have to kind of take steps back and then make them into, you know, cells that produce dopamine. There's a worry in that number one, it sounds like the cost in the end is $1 million or pounds, right? It takes a lot of, even for one patient to be able to do that process, that is going to be, you know, cost prohibitive. But also, I guess, could you speak to a question that I have is, you know, if those cells are already programmed to become Parkinson's, let's say we think that there's a genetic or some, you know, nurture versus nature is the sense of what causes Parkinson's, but the nature of those cells is to have, you know, something that ends up becoming dysfunctional. How do we know that we're not going to end up with dysfunctional cells once we kind of do all that reprogramming and stuff?
Yeah, I mean, it's a very good question. So just to sort of recap, so the fetal dopamine cells, you're absolutely right. I mean, the logistics of it, you need at least 4 fetuses, 3 to 4 fetuses per side to graft a patient. So logistically, it's not possible. Obviously, ethically, it's a very contentious area because the only cells you can use are from women who've decided to have termination of pregnancies, abortion. So, you know, different countries take different views on that. So there are major ethical aspects as well as logistical. Having said that, it's also important to know that some of the fetal transplants have acquired the pathology of Parkinson's disease in them years after they were put in. So this has raised the idea that the protein causing Parkinson's disease spreads into the transplant. So these unrelated cells do acquire the pathology of Parkinson's disease. So coming back to your question, if you take cells from the patient who already has declared that they are likely to get Parkinson's disease and have it, the cells you are producing therefore probably have a greater risk of developing it. Given unrelated fetal tissue has done it, there is a worry that the autologous, i.e. the patient's own iPS cells, will develop the pathology and therefore the transplant will stop working more quickly than if you take unrelated embryonic stem cells or unrelated iPS derived cells. It's also fair to say, as you point out, that at the moment the process for making a cell therapy is regulated around the product you make. So if I take, Rebecca and make dopamine cells from her iPS cells, I would have to do a full testing battery, safety, everything. That would cost several £1 million, or several $1 million. They're all about the same these days. And treat Rebecca. And then if I want to treat India, I have to do exactly the same again. So as it stands, it would not be financially possible. Now, it could be that the regulators will regulate the process, not the product, in which case you can say that I can always do it the same way. Then the cells become much cheaper. So that's why autologous is not particularly attractive. Then the question comes, do I use unrelated iPS or ES? Some people don't like embryonic stem cells because you have to destroy an embryo in order to make the cell line, so there are ethical concerns. Human embryonic stem cells have been around for since 1998. Human iPS cells have only been around since 2007. So they're still relatively new, and you have to engineer iPS cells to become iPS cells. So there are worries about whether you've actually modified their behavior, which makes them more dangerous. We have evidence for that. So most of this is theory, but at the moment, I think most people go for iPS over ES commercially, because what they actually think is that the embryonic stem cell problem, especially within the ethics in America, would put people off.
I see. Okay. There's a lot of question about cord blood banking, and this is like a big deal here. Could you speak to that and whether there's any utility in patients with Parkinson's for doing that?
No, I mean, it depends on what you want to do. So there are tons of stem cell therapies out there, which I slightly glossed over. What I was talking about today was taking stem cells, turning them into dopamine cells and transplanting them into the brain. There are millions of, well, not millions, there are lots of other types of stem cells, so umbilical cord, mesenchymal stem cells, adipose stem cells. All of these are said to be useful for these conditions. I have no idea how. They sort of produce something that's magical that makes things better. In some countries, you can have them injected under the skin and they'll make you better. No idea how they get into the brain. So there is a lot of nonsense out there, really. It's not to say that they couldn't be used eventually to try and help people, but as it stands at the moment, none of them would be competitive. The only therapies that are worth pursuing, I think, in the cell-based field are those which are designed to replace the dopamine cells by making dopamine cells with that stem cell source.
Okay. That makes sense. So one question was about, when we're doing these trials, a lot of the times historically, we've taken patients that are farther along in the disease and tried to do these therapies. And I think that you're showing, these slide at the end where, you're sort of backing up to before we even really, are needing much symptomatic treatment for Parkinson's. We've had a lot of speakers on talking about, we had Matt Stern on actually earlier, talking about the, sense of, this Parkinson at risk kind of population, and can we figure out, who has premotor Parkinson's? Could you speak a little bit to that? And I think I also have a second question that I'll ask you after that.
Yeah, it depends what you want to do. So I think one of the reasons which the fertilizer trials have failed, the growth factors, the GDNF, is because you've taken people at a point when all the roses have gone. So So however good your fertilizer is, nothing's going to be rescued. So so I think there's a sort of major reason for taking early stage patients with growth factors if you're going to repair the brain. Cell-based therapies you could use at any point, to be honest, because you're just replacing the lost dopamine cells. And in the early trials, you'll have to take people who are relatively advanced, just because the ethical boards would be keen for you to take patients who have a lot of therapeutic options available to them and give them an experimental treatment. But I think ultimately these cells, as I said in the final but one slide, could be used as a first-line treatment if they're proven to be safe and efficacious in bigger trials with more advanced patients. I don't think you need to use them in prodromal Parkinson's because, as most people will know, especially those on the call who have Parkinson's, when you get newly diagnosed Parkinson's and you're stuck on a bit of L-DOPA, I mean, you pretty much go back to normal if you get the dose right. So, you know, we don't need to treat people before they've got any motor features because that's what we're largely targeting with the therapies. So I think for this, we wouldn't need to go to prodromal. It's also fair to say that I think just a point that's worth making is none of these therapies are mutually exclusive. So if Matt Stern has a disease-modifying therapy that slows down the disease in prodromal PD, then you could start someone on that when they develop the first motor features, we stick in our cells and the two together can work together. So I think the combining therapies is we should be thinking of. It's just the regulatory bodies get a bit excited if you do more than one thing in one patient at one time.
Yes, that makes sense. But I think patients are hungry, you know, there's a sense that we shouldn't be doing this ethically because patients are so mild. But some of the patients that are mild are like, you know, sign me up for these trials because I want to do whatever I can. So there's a sort of, you know, balance between advising people about some of these things, especially in therapies that may have, you know, side effects and are involving brain surgery, you know, so so it becomes a hard thing to balance. There's been a question about whether these cells could produce other things. I mean, one of the questions is, you know, can they cause cancer? Is there like a likelihood of tumors being generated or things like that?
Yeah, I mean, that's the big worry. So the big worry is that obviously, you know, embryonic stem cells have the potential to make any cell in your body. So every single person on this call was an embryonic stem cell at some point in their existence. So that's what we all began life as. So they have the potential to obviously turn into any part of us. So that's one problem. The other problem is, of course, that if they start growing as stem cells normally do, then we're in big trouble because they will just form tumours. I think whilst that's a theoretical risk, there's no actual evidence from that, from the trials or the data we have today. So the differentiation protocols, the protocols we use to turn stem cells into dopamine cells, actually that protocol, which is quite efficient and only takes 2 or 3 weeks, is very unfriendly for growing stem cells. The way of killing off stem cells is actually using the differentiation protocol. So that at the end of it, you have cells that are committed to being dopamine. The stem cells have actually died off because they don't like trying to grow in that environment. And so the group in Sweden, Marlin Palmer's group, have transplanted their cells in over 1,300 animals, and they've never seen a tumour in any of them. So that is, of course, why we have to go slowly at the beginning, because that is the major anxiety from the regulatory authorities. Actually, we've never seen a problem with that with the protocols that we have now.
Okay. That makes sense. And then there's a few questions about immunosuppression, what the need is to take that if you're taking, you know, the embryonic versus your own cells. What's your sense of that?
Yeah, so if you take, so the advantage of doing your own cells is that in theory they're just your own cells in the brain, so you don't need to give any immune suppression, so your immune system will see them as part of yourself. That is still not entirely proven that if you make stem cells from yourself, they're still recognized as yourself. So there is some question about whether they're rejected. But for most cells, which you put in the brain, you probably will need to give some immunosuppression, but not much. So in the fetal transplant, some people have put fetal cells from unrelated donors into the brains of patients with Parkinson's, not giving them any immunosuppression, and the cells can still be found years later. So cells can survive without immunosuppression, they're just not surviving as well. So our belief is that you would need to give some immunosuppression for a year or two, and that's probably all you would need to actually do. So this is not like having a kidney or a liver or a heart where you need to be on long-term heavy-duty immunosuppression for decades. You would probably only need a short course for a couple of years, and that pathology I showed you of that transplant 25 years after it was put in, that patient had not been on any immunosuppression for 20 years.
There's a number of questions about cost, what your guesstimate, I guess we said at least £1 million for the ones where we're taking our own cell, and like you talked about Rebecca and me. But what about the other, what's your sense of if the embryonic stem cell surgeries worked? And I guess you have this trial that's ongoing that we'll hopefully get some information about in the next year, right? Or something? Yeah. What do you think the cost would be?
I think it'll be quite cheap, but the companies will make it expensive. I mean, if I start, so for example, if I start with a 6-well plate of embryonic stem cells, so something that's about that big. So if I went into the lab today with a plate of six wells of embryonic stem cells and used the protocols, by the end of this month, I would have made enough dopamine cells to treat 300 people. That's me. And that's in 16 days. So I have 1,000 plates and a robot. That is whatever that is, 300,000. So that's going to be most people with Parkinson's, at least within Europe. So that's one production run in a GMP facility, which is quite expensive, but it's less than 3 weeks. So actually the cost of making the cells is not going to be prohibitively high. There's obviously the cost of the surgery, and there'll be a cost of immunosuppression, but the other beauty of this system is if it works, it's a one-off treatment. So it's not like deep brain stimulation when they have to change the battery every few years. It's not like the current treatment of Parkinson's where you have to take drugs every day. It should actually be relatively cheap. Now, the question will be, what's the final price? And the final price will be determined by the people who make them, and that won't be me.
Well, I hope you get some for spending 30 years of your life working on this. I hope you get something.
I'll probably get a free vial.
Or some cooler paintings too. But you have some amazing stuff in that room back there. Is this your house?
It is my house, yeah. Because we're encouraged not to go to work if we don't have to. And since I was on for the weekend in the hospital, I've had today working at home.
Well, we appreciate you taking the time, and that looks like a museum. So, we'll have to have another one of these where you walk around your house and teach us about all the paintings. They look pretty fabulous. So there's been a few other questions. So one is, so when we transplant cells, these A9 cells, how many of them actually live?
Yeah, it's a good question. So actually you don't need that many to survive. So one of the other reasons Parkinson's is so attractive, apart from the fact it's one cell type with one chemical, is, as I said, you have 500,000 dopamine cells on either side of your brain. When you lose half those, 250,000, you get features of Parkinson's. Now, we put the cells not where they're lost, but where they work, and you probably need to get about 100,000 surviving in order to have benefit. So at the moment, that's something we can easily achieve with implanting 1 million, 2 million cells. We'll easily get 100 to 200,000 surviving cells. So that's why it comes back to this sort of question of cost. Because you don't need large numbers, you can actually produce enough very easily. So, not many.
There's been a comment here that the reason you look so young is because you use cell therapy, apparently. So you have a lot of fans here with who are commenting on how you don't look like a fogey, you look like a fellow. So you must come back and talk to us, because you have a lot of people who care about you here, which is why I do this, because they're very sweet.
No, it's the injections of detergent I've been giving myself for years that works.
Fabulous. So a couple more questions. So we talked about the dopaminergic system, right? And replacing cells that produce dopamine. We've had a number of your colleagues, Ray Chowdhury, a number of other folks from the UK teaching us about non-motor symptoms of Parkinson's. So it's interesting that the quality of life measures, and you see the motor improvement, but then there's a significant, what I would say almost larger than just the motor improvement benefits in these other areas, right? So what do you think that is?
Well, I think there's two reasons for that. One is, I think, if you make people motorically better, there are lots of non-motor symptoms which are related to poor motor control. So the obvious one is if your motor control is very poor and you don't sleep very well at night, as a result of that, you will have fatigue during the day, you'll have a degree of depression, there'll be all sorts of issues you'll have, and that's related to poor motor control. So if you make the motor control better, those non-motor aspects will get better. That's the first thing. Secondly, some of the non-motor aspects of Parkinson's have a dopamine basis. So, for example, when people have problems multitasking, which is quite common in Parkinson's, that is almost certainly due to a problem with having a dysfunctional dopamine system in the striatum. So one of the interesting things, which I haven't talked about, but we're quite interested in, is in early Parkinson's, dopamine levels drop in the deep parts of the brain, but they're super high in the front part of your brain. So actually in early Parkinson's, dopamine levels in the front part of your brain are 2.5 times normal. So actually the difference in dopamine levels between bits of your brain affects your cognitive functioning, how well you can do memory tasks and thinking tasks. So by putting dopamine back and normalizing in that area, we think will improve some thinking tasks. And by improving the motor tasks, will improve some of the non-motor aspects of it.
Wow, that's really interesting.
There's obviously lots of pathology elsewhere, and that mediates some of the non-motor. But I think, you could see in that one patient in the New England Journal from last month, his quality of life went from pretty bad to normal.
And so how much is that placebo effect? What's your sense of, because these are...
I would think it's quite large because he paid for it.
I mean, that is the problem with a lot of these trials, especially open label, right? And these sort of one-off sort of, people that are reporting things, it becomes, hard to know what is actually.
And so some people have criticised me when I show that video saying that could be a placebo effect. And I say, well, if that's a placebo effect 20 years after you put it in, I don't really care. I mean, that'll do. I mean, but, the thing which puts you off it being placebo effect is the length of time. And the fact that if you look at dopamine scans in these patients who've done extremely well, they're normalized. So the dopamine levels come back normal. So it's quite hard to get away from that. But it is a problem with the field. And of course, the very controversial area is how you do trials with these cells. So this side of the Atlantic, we're very keen on we're much more keen on open label studies. In the States, they're much keen on taking people and drilling holes in their head and pretending to do surgery and not giving transplants. So-called, sham surgery. So how you do the trials and work out whether you've got a true effect versus a placebo effect is difficult.
Yeah, that is interesting. But I'm glad that you're getting together and having these conversations. And it sounds like by and large, most people are collaborating quite well. I want to be on the...
Well, it is. I mean, it's getting a bit more difficult now because everyone's linked to companies, and companies tend not to like talking to each other unless it's a COVID-19 vaccine.
Right. I'd like to be on G-Force PD just so I can list that on my CV, and it looks like a beautiful meeting. So please sign me up. One other question, and then we'll get maybe a little bit more into the ethics of stem cell tourism and things like that. So one question I have is, for the genetic forms of PD, which are rare, right? They're not as common nearly as what we see in bread and butter, things that walk into our offices. What's your sense of that as a target for some of these therapies?
Well, it's an interesting question because people follow different courses depending on the genetic form of it. So there are certain genetic forms which probably you progress more quickly. So people who carry mutations in the Gaucher's gene, the GBA gene, tend to do less well. So they would not be a good candidate for cell-based therapies. Now, people who have Parkin mutations, so the younger onset patients, often have a very slowly progressive disease, very responsive to dopamine. And you could imagine that they, in essence, could be cured by one of these cell-based therapies because they have a very purely dopaminergic pathology. They don't tend to get Lewy bodies. So I think certain genetic forms of it may be more amenable to it. It's difficult with others because things, as you say, a lot of these are very rare. So the LRRK2 mutations, they can present in any way, and I can't think why they wouldn't respond to these cell-based therapies. People generally speaking with genetic forms look to treat the gene defect because it's a clear target. And so people are more interested in using, genetic manipulation or specific drugs targeting pathways linked to that gene. Yeah.
So I think let's move on a little bit to some of these other sort of stem cell tourism and, sort of your sense of how, as a patient, you can approach sort of and people have written in the comments here that like every day they're spammed and there's like a barrage of stem cell stuff, maybe after they sign up for some Parkinson's courses, somebody out there in the world is spamming them every day with, you know, all this stuff. And I think we've talked a little bit, I have an interest in integrative medicine, and I've been really thinking about, you know, how to help patients in this, you know, sort of borderland between things that are you know, Western medicine, but also some of these other approaches. And a lot of the worry is that when people Google Parkinson's or the stuff that they get in their feed, that they tend to, you know, sort of, even if they don't want to, you know, fixate on, there's just a barrage of hype out there and people trying to, you know, approach our patients and sell them something or have them sign up for something. So maybe you could speak a little bit to that in the stem cell world. And, you know, I think you probably have a lot of experience with, you know, people coming to you and saying, you know, how about Mexico or, you know, Taiwan or wherever. And they're, like you said, they're taking cells from somewhere and sticking them somewhere, which, you know, you're not even sure what's happening. So maybe you could speak a little bit to that.
Yeah, I mean, you know, it's easy when you don't have the condition. So, you know, it's easy to sort of take some moralistic high ground and say you must never do this. But I do say that with sincerity, because people have done badly with these therapies. So in that same journal, the New England Journal of Medicine, they had a paper about 2 or 3 years ago of people who had a different condition, age-related macular degeneration, so going blind, went down to Florida, and adipose stem cell injections into their eye to repair their age-related macular degeneration. They went from poor vision to no vision. There's somebody in New York who had spinal cord injury, had a transplant of stem cells, I can't remember where, now has a spinal cord tumour as a result of those stem cells being put in. There was a child that went to Russia with a rare neurological condition, had stem cells injected, died of disseminated tumours throughout their brain and spinal cord. So if you go to places where these are not regulated and there's no obvious science behind what they're doing, then you can end up being worse than you actually are now. And sometimes people find that hard to believe. And for most stem cell therapies that are out there, the major harm will be that you've lost a large chunk of your bank account to them. But my own advice is that you should never pay for a stem cell therapy at the moment for Parkinson's disease, because none of them are proven. So all of them should be subject to a trial. So if someone is offering you a stem cell therapy, which you're having to pay for, there can be no reason for that. It's not like, I don't know what it's like in the States, but, you know, if you want to have a cataract removed, everyone knows if you have a cataract removed and a lens put in, your eyes will be better. That's proven, that's well documented. You want to pay for it because that is a perfectly legit treatment. This has never been proven. So paying for any stem cell therapy, you must never do. If you are approached and you want to ask questions about it, the International Society for Stem Cell Research, the ISSCR, has a very good website, which we've tried to make very patient and public friendly, with lots of questions you can ask those companies about what they're doing, or ask yourself about. So I think trying to get to the essence of what they're trying to do and whether there's any value in it, what you'll find is often these companies say, well, actually, it's all a company secret. That's why we don't publish on it. We can't tell you about that. That's all, you know, related to our IP and everything else. Don't believe it. If they've not published on it, it can't be true. So, you know, if you look at all the major therapies that have actually come through in medicine, they've all come from places which have published on it and have been the subject of peer review. So it's a great temptation. There are lots of people offering it, but actually there's no evidence that anything out there at the moment for Parkinson's disease with stem cells is worth having.
Okay. I mean, I think that the bottom line is that it's important to research things, to be communicative with the doctor that's treating you. Rebecca did put the, it's isscr.org, and we'll link that with this talk as well. And, you know, you can set it around to your friends, and hopefully, you know, people will be able to watch this talk on YouTube afterwards. And so I think really it's about being very kind of cautious, especially when you have a company that's working on their own. When you see this, you've really covered you showed us a map of the world, who's doing what, where, and, you know, I think that you've done this for 30 years. And so for you to be this rigorous and this systematic and still be so cautious, I think it speaks volumes to me. And I really commend you for your, you know, hard work over the years. And, in this area. So maybe in the last few minutes, you can tell us, we have about 5 minutes left. I always like to end with some silver linings, some, positive hope and a message, to the group here, which is largely patients and caregivers from all over the world, but people watch this video later, and, hundreds of people are watching these videos now, sending them around to their friends. And we're just trying to connect some people, through this, global pandemic. But I think it, hopefully will be, a video that we can share as sort of the state of, state of the union of, this issue, now in 2020. So maybe you can speak about the future and some hope and, what you really see is, since you've been a doctor in this field for now, it sounds like almost 3 decades working on this. So tell us a little bit about what you think.
I think with particular respect to cell-based therapies, I would say if I was giving this talk 10 years ago, there was no protocol for making stem cell derived dopamine cells from human stem cells. So I could offer you no hope of a treatment because there was no protocols. Fetal tissue had been shown not to work at that point, so there was no interest in anybody doing it. So in the last 10 years, this field has moved forward scientifically, and we're now, as I say, on the cusp of clinical trials. So that is a great advance. The involvement of companies brings with it challenges, but it also brings with it great potential. So as I say, we work closely with Novo Nordisk in Europe. Novo Nordisk is the 4th biggest pharma company in the world. If this works, this will move at a very fast speed into clinic. So it won't be left to academics like me to try and find another, £5 here to get this going and that. This will suddenly accelerate, and this will become a major product for everybody if it works. So the infrastructure for translating this into a major therapy like deep brain stimulation did in the 90s is there. So that, I think, is hugely exciting. I think also in my time in the last 10 or 20 years of Parkinson's, the new treatments being tried in 2020 for Parkinson's is hugely different to where it was. We didn't really have much going on in the early part of this century. Now we have all these vaccines, we have all this drug repurposing trials, we have better deep brain stimulation, we have the cells I've talked about, we've got gene therapies replacing dopamine. There are lots of things going on. So I think there's huge potential for better treatments for this condition. And as I said, I think combining treatments will be a very important way forward. And I think ultimately, so my ultimate goal when I retire is that cell-based therapies will be a treatment for everyone with Parkinson's disease, and that will now be trialled with disease-modifying therapies to see if we can actually improve the efficacy of our transplants while slowing down the underlying disease.
Well, I really hope that happens. I mean, it's such an exciting field and you've worked so hard. Maybe in the last minute, Professor Parker, you can tell us a little bit about how patients can advocate in this area for themselves, because we have a lot of very, sort of activist type patients on this, and I think other people will be watching as well. So how can they help move this field forward?
So I think the key thing is actually to get engaged with the organisations which are out there already. So I think a lot of people obviously get a bit disillusioned with them. I have a lot to do with the World Parkinson's Coalition and the World Parkinson's Congress. Absolutely terrific. I think sign up to that because they have huge persuading powers. Link up with your local research clinics. Because you will have a major impact when we try and get things through ethics boards. We try and tell people, we're trying to get the regulatory authorities to help us. We can talk as much as we like. Patient comes along and says something, they fall over, because you are the people that determine what's going on. And there's this sort of slight belief that the doctors and scientists are trying to pull a fast one on you, rather than actually you're the ones who really want this change. So I used to have a lot to do with the late Tom Isaacs in the Cure Parkinson's Trust. And Tom and I used to go and give lots of talks together, and it was all about working together to try and change things. And patient voice has much more weight than any of us.
I think that's very inspirational, and I think that's absolutely true. We want to have the regulatory boards fall over with the patients influencing them. So I think that would be exciting. Well, thank you so much for taking the time in the middle of your, crazy work week and showing us some of the art in the background there. It's just been a sheer pleasure to get to know you a little bit through this and to learn about this work. And it's just been a very clear, such a beautiful talk you gave. I really, feel like I've learned a lot. So I really thank you. Somebody wrote Tom Isaac rocked. So,
great man.
So anyway, thank you, Professor Barker, and enjoy the rest of your evening. And we appreciate you so much. And Rebecca, we'll send it back to you for our goodbye wave.
Yes, and thank you so much, Professor Barker. I mean, if you could read these, I mean, it just keeps going with how much people have really enjoyed this talk today. And for those of you who are really loving this series and getting to see Dr. Subramanian, we have a packed month this July. I mean, I know it's sort of in the middle, but we have 2 additional programs this week in this specific series. We have one on Thursday as well as one on Friday. I'm going to put our link in the chat. So if you go there, you can see what we have upcoming, and we hope you will join us. So I am going to sign us off. If everybody, if you can turn your cameras on, we're going to do our wave goodbye. We love seeing you and having you join us, and I hope you have a wonderful week. Bye.
Thank you.
Thank you.